exciting to be here to talk about some of our work on comparing sex differences in athletes at the molecular level. So I'm going to start. I hope you all recognize the central dogma setting the stage for this molecular part of adaptation. So we are studying, everything from the genetics, so the DNA, the blueprint, and the copies of the DNA, so the RNA, that then get translated into proteins, so the functional molecules, and again, these proteins can also be modified. So these are all different levels of molecular regulation that we can study in exercise adaptation as well as in athletes. So today I'm going to focus on the transcriptome, which is when we take a biosample and we look at all of the transcripts that are present in that sample at one time, as well as the proteome, so looking at all of the proteins in that sample. And we study multi-omics, so we're trying to build a molecular map of the adaptation to exercise and what happens with athletic performance. And the goal with studying multi-omics is really to get a more comprehensive picture of what happens across these different organs and across different levels of molecular regulation in response to exercise. So building this fuller picture of what happens in our bodies when we train. And we can do this in different ways. We can study the extremes of human performance. So we can take the athletes that you have on the right, so these highly adapted people, and we can compare them to untrained sedentary men and women. We can also look at training adaptation. So we can start with people who are sedentary, and we can go towards a more trained phase and look at before and after training and what happens at the molecular level. So does training affect men and women equally? What happens molecularly when we train? That's what I'm going to briefly talk to you about today. And I'm going to focus on skeletal muscle. So what's interesting with skeletal muscle, if we take sedentary men and women and we compare their molecular profile, so the transcriptome and the proteome, we see quite large sex differences. So the muscles are very different for men and women in the untrained state. And we've seen this repeatedly in multiple studies. However, if we look at endurance athletes, so we take muscle biopsies from these highly trained individuals, these differences are reduced. So it seems that the molecular adaptations, so the functional demands of training, the metabolic demands of training, drive muscles to become more similar between men and women in athletes. If we look at rats and we train them, we see similar things. So here we're looking at the proteome, and here we're not looking at individual proteins or transcripts, but rather these molecular pathways. We call them pathway enrichment analysis. So these are some of the key, mostly metabolic functions that are different between sedentary rats. So the first row there that you see with a lot of red dots show that there are differences between male and female rats in the sedentary state. But if we put these rats on a treadmill and train them for 8 weeks, most of these differences disappear. So there is a convergence of skeletal muscle with training in both humans and animals. But the plot twist here is if we look at white adipose instead. So this is white adipose tissue from sedentary people as well as from endurance athletes. So there you can see there are still quite big differences between men and women in the sedentary state. So a couple of thousand genes differ in their transcriptional profile in sedentary people. But what's super interesting is in athletes, so if we take white adipose biopsy samples, this is from abdominal fat from endurance athletes, we see that these differences become even more prominent. So white adipose seem to have a more different function in supporting male versus female athletes from a metabolic standpoint. So to summarize, some of these tissue-specific responses really challenge this one-size-fits-all training recommendations that we all tend to receive from everything from American Heart Association to American College of Sports Medicine. So we really need to start thinking about, of course, consider sex when we design different training recommendations, but also maybe what type of tissue we're trying to optimize. What functional adaptation are we looking for as we design our training programs? So the one-size-fits-all model doesn't really work. And beyond the finish line, we see that molecular adaptations for similar function and similar demands looks very different at the molecular level. So with that, I want to thank you for listening, and also, of course, thank the Tsai Human Performance Alliance, as well as some of our other funders and great collaborators. Thank you very much.